Yeah, Charlie?
Oh yeah. I guess anywhere.
Do you want someone to hold this, or is it okay to just?
Oh, Roger. I brought mine too. You have one.
Do you want me to just leave this here? Just stand there.
Just give it to Jim.
That seems like a lot of 45 minutes.
That's true. Oh, if Dan says it works, okay.
Okay.
Perfect. Well, Jim, do you have a laptop anymore?
We like comments on the live stream. Yeah.
PhD students' favorite SKCB comics. Oh, there it is. Watch the tape.
Alright, I gotta keep down, yeah.
I like the
64, that should be.
MIT time is like 5 minutes, right? So yeah.
So that's wrong?
That's a live stream because a couple of my friends
Oh,
watch
I
that.
see.
Ron is theoretically coming. I mean, he emailed me earlier today. So. I'll see. Should I go try to find him?
Yeah, it's my hands are very, very dirty.
Okay, that was one. This is a different angle. One more angle.
Thanks.
Yeah. Oh, the Thesis for
Yes.
no, I almost had to write it. I almost forgot that. Sorry. I sent the one. Yeah. I brought the. I don't know. So Andrew said she got the signatures day of. This is not on like the right paper, but I'll just ask her soon. I don't think it is. I think you can get that side. Oh, okay. How do you know that? Because I have no idea. I think it's just this, and then they say you're going to share the email. Yeah, there's.
Or
I still figuring you don't need it while you're presenting. Nope, I'm good.
Doors. You guys close the doors.
It's an ecstasy.
Hi, everyone. So welcome to the Thesis Defense of Vyas. As you know, he's joined our lab in 2012.
Officially, yeah.
He did his undergraduate work at Penn with Jason Burdick, who called me up personally and said, you must admit this person to your lab. And then I called Jason a month later and said, you were right. And it's been a pleasure to have him. Together with Andrew, who recently defended, this is our first example of roommates in the same lab. Going all the way through in lockstep. And I have to say, I was skeptical about how it would work out, but they seem to have come out the other side better than the sum of their parts. So it's really been terrific to have them in the lab. They work in really different areas. Vyas, as you're going to see, works virology. Charlie Rice, who is our committee member virologist, is actually not here today, but he's been very connected to all the work. So no further ado, I welcome him.
Thank you. So for all the probably defended a couple weeks ago, and sent me this unbeknownst to me as a way to prepare me for a thesis defense. Hopefully I won't have to use it. So yeah, thanks to everyone for coming to my thesis defense talk. The genomic profiling and perturbation of hepatitis B virus infection. Thanks a lot, Cynthia, for the warm introduction. And Rob, my thesis reader and committee chair, for being here. Charlie, as Sangeeta said, unfortunately couldn't make it because he's off in the wilderness in Wyoming. I think the last interesting virus he started working on was found in a rat in New York City, so one can only imagine what crazy things he'll find on this trip to start working on. So I wanted to start with a little bit of history. So the 50 years ago, Baruch Bloomberg reported on the discovery of the so-called Australia antigen, which was in the blood of an Australian Aboriginal and a hemophiliac who had been transfused. This was really the first report in 1966 of the work that would then become the body of literature on hepatitis B, which over the last 50 years has the development of effective screening tools, an effective vaccine, and public health campaign. Unfortunately, as you'll hear in this talk, not to the development of drugs that are effective enough to actually clear the virus from patients who are chronically infected, for the most part. However, this initial report was actually enough to catalyze the field to microscopy analyses of these virions that you can kind of see here in infected hepatocytes, leading to the development of the first approved vaccine only 15 years later in 1981. As another interesting historical aside, when the vaccine was initially developed, it was produced by purifying surface antigen particles from hepatitis B virus infected patients. And unbeknownst to everyone at the time, a lot of these patients were also HIV positive, and this was a virus that was not detected at the time. Luckily, the activation of 80 or else the Hep B vaccine would have actually led to the spread of HIV, which is kind of an interesting thing. Baruch Bloomberg won the Nobel Prize 10 years later for his discovery and his role in the discovery of hepatitis B virus, and one other historical science I think is interesting. In his Nobel acceptance speech in 1976, he actually had some prescient commentary or advice. We're quoting, "On a broader level, the ethical issue is raised as to the extent to which biological knowledge about individuals should impinge on our daily lives. Is it appropriate to regulate the risks inherent in people living together and interacting with each other," which could be said just as much about, for example, genetic data today. It's kind of an interesting thing to think about. So from there, we luckily got to the point where a vaccine exists and therefore have been able to eradicate a lot of hepatitis B in around the world, in both the developed and developing world. But as you can see briefly here from this epidemiological map, there's still a high level of incidence in East Asia and in a lot of West and Central Africa, leading to a cumulative incidence of over 300 million people around the world being chronically infected, which due to the end-stage liver disease that can result leads to about 1 million or more deaths a year. And unfortunately, although it's kind of a poignant fact, the combined mortality from hepatitis B and hepatitis C now actually exceeds that from HIV/AIDS due to the increased penetrance of antiretroviral treatment. So what happens when HBV infection actually occurs? So typically, when adults are infected, they almost always clear the virus in the acute phase. About 1% of them progress to chronic infection. When children, either perinatally or through the age of 5, when the immune system is underdeveloped or infected, they go on to progress to chronic infection at a much greater rate. And once there is chronic infection, it kind of proceeds through these stages of viral hepatitis. There's the immune reactive phase, which is actually this chronic hepatitis phase, which can lead to the establishment of an inactive carrier state, which only turns back on in the situation of immunosuppression, but often over decades will lead to cirrhosis and to either cirrhotic decompensation or hepatocellular carcinoma, which is an imminent cause of death in a lot of these patients who died due to hepatitis B virus infection. And unfortunately, although the vaccine exists and is extremely good from a public health standpoint, it's very safe and effective, the full penetrance of immunization is less than 80% around the world for children, which leads to a lot of perinatal transmission and no ability to break the transmission cycle here. So just to get a little bit into the molecular biology of the virus, this is a lot of information which we don't fully need. The most important parts are to say that hepatitis B is a very small, partially double-stranded DNA virus. It has a genome of only about 3,000, or a little bit greater than that, bases, and encodes for 4 open reading frames, which overlap here. So there's the surface antigen, which was the Australia antigen that kind of is generally immune suppressive and that the virus uses to enter into hepatocytes to infect them. The polymerase, which has both reverse transcriptase and DNA polymerase activity. The core protein, which actually makes up the capsid here of the virus. And the X protein, which is named partially because it was kind of mysterious for a long time what it does, and it serves as a promiscuous transcriptional activator of a variety of transcription factors in the cell, and actually leads to a lot of the host pathogen interactions that we'll discuss a little bit. So intracellularly, just to go briefly over the life cycle and the important parts of HBV, it's a little complex, but try to simplify it in the schematic here. So it enters through its receptor called NTCP, or the sodium taurocholate cotransporter, which is actually a bile acid transporter. The virus enters into the cytoplasm, gets shuttled to the nucleus actively, and this is where this DNA here, which is partially double-stranded, actually co-opts the host endogenous DNA repair machinery in order to form a fully double-stranded nucleosome-associated cccDNA, which is kind of a tongue twister in a word. It stands for covalent closed circular DNA. But this is the viral species that is so hard to eliminate because it just sits there episomally and has a half-life of decades, which makes it so hard to clear this virus. From there, there's transcription of the viral RNAs, leading to protein production and production of the viral DNA from the RNAs. And the other thing that I wanted to mention here is the way that hepatitis B virus infection is currently treated. So these treatments are in general not curative. They're nucleoside analogs which block reverse transcription here, or interferon, which is a general innate immune activator which can help to clear the virus. But even interferon, when treating for about a year or more, results in functional cure rates, which is not even a complete cure, of only 3 to 7%. So extremely low. So to get into what the body and content of this thesis is, overall we looked to do 3 things. The first was to develop primary model systems for hepatitis B virus infection. The second was to use these model systems to profile and perturb the host response as a way to try to find viral weak points based on how it interacts with the host liver. And third was to use a novel genome editing platform to actually directly attack cccDNA and to clear viral infection. So to start out with the first aim now, in general, the models that exist for studying hepatitis B are not great. Ideally, what you would want in a model system is for it to be easy to use, for the true life cycle and host virus interactions to be recapitulated, but also for it to be experimentally tractable, so to have low enough cost and high enough throughput to be able to try to answer a lot of biological questions. So the original models that kind of existed were small animal models for viruses that were similar to hepatitis B, so they don't fully really capture the real life cycle because they're important variations. And interestingly, the Tupaya, which for some reason is infectable with true human hepatitis B, which otherwise only infects chimpanzees. Unfortunately, since these are not exactly model organisms, these experiments are also extremely expensive to do. There are Hep B producer cells, which can produce the virus and are affected by viral proteins, but don't actually get reinfected, and therefore it's of limited utility in really studying the biology. And newly, since the discovery of the Hep B receptor, which I mentioned, there's been a knock-in of this receptor to create Hep B susceptible cell lines, which unfortunately are dysregulated in a lot of the biology that's actually interesting when it comes to things like cell cycling, which Hep B affects, leading to difficulty in interpreting those results. And then there are human liver chimeric mice, which have human hepatocytes in a mouse liver. They're actually great for studying this, but they're extremely expensive, hard to use, and don't especially have good throughput. So the ideal thing we'd want to do is to have in vitro primary hepatocyte models, primary human hepatocyte models of studying Hep B infection. And so when people have tried to infect primary human hepatocytes in the past, what you see, this is looking at surface antigen production over time, is there's rapid decline post-seeding in the infection efficiency of the production of viral protein, which correlates with the decline in the functional capacity of these hepatocytes, which is something that Sangeeta has actually been working on kind of reversing even from the time of her PhD. And so since then, we've developed a system by co-opting techniques from semiconductor industry, micropatterning, photolithography, things like that, to create a primary hepatocyte model system where hepatocytes are stabilized in their function over time. And what this has allowed us to do is to actually study other infectious diseases as well as some other metabolic diseases now. So here are some examples of seeing productive infection in malaria and in HCV. So what we wanted to do here was, when we started out, to try and use these micropattern co-cultures to develop a hepatitis B virus model system. So to do this, the first thing that we did was to look at the actual expression of the receptor here, which, as we know from the hepatocyte knock-in cell line experiments, is extremely important for infection. And we compared these MPCCs, they're called, to randomly co-cultured hepatocytes with stromal cells. And we saw that the receptor expression was much higher in these primary hepatocytes. So after doing some optimization, we were able to, using patient-derived viral isolates, productively infect hepatocytes. And this is, the surface antigen is a measure of viral protein secreted. This is a measure of viral RNA transcription. As you can see, it's much higher in micropattern versus random co-cultures. And very importantly, we were able to detect pretty significant copy numbers of cccDNA, showing that this is actually a true productive infectious life cycle. And in some immunofluorescent analyses, we see that core protein is localized correctly and that we're actually getting true infection. So what we wanted to do here was then to use this system now in primary hepatocytes to ask some questions that have been kind of ambiguous in the literature throughout. So one of these questions is whether hep B actually does stimulate an innate immune response in hepatocytes, in these liver cells. And so this innate immune response is driven by a set of proteins called interferons, which essentially are turned on by epithelial cells when they sense danger signals coming from, say, viruses or bacteria or other potentially pathogenic material, and lead to the production downstream of effectors that can restrict this response, either by directly attacking viruses and viral nucleic acids or by modifying the host. So what we saw here is that we did get significant upregulation of a number of interferons throughout this time course at various degrees, and also the upregulation of effectors of the interferon pathway called ISGs, or interferon-stimulated genes, after this. So this induction was not as strong as what you would see in, for example, hepatitis C virus, which is canonically a strong inducer of innate immunity, but it still was quite significant and pretty robust. What we wanted to do to check that this was actually due to infection, since we were using infectious human serum, is to then block infection using that same inoculum, using the reverse transcriptase inhibitor entecavir. And what we see is that when we do that, we largely aggregate the induction in these ISGs over time. So based on this data and the fact that we realize this infectious system was actually inducing the innate immune response, we wanted to see if we could boost infection in order to increase the dynamic range for developing an antiviral testing platform by actually inhibiting this innate immune response. So that's what we did by using a broad spectrum inhibitor of JAK-STAT signaling to block the signaling downstream of interferon. And by doing that, as you can see from a number of viral readouts, we were able to strongly boost the robustness of infection and actually see an increase in the size of the cccDNA pool over time. So now that we had that, we wanted to look at whether we could actually use these MPCCs as an antiviral testing platform. And so as you can see in panels A and B, in a prophylactic mode where we start with treatment before infection, and in C where we start a week after, we're able to actually get, with two different antivirals, two different mechanisms of action, the two that I showed before, you're able to see reductions in a number of viral parameters. And interestingly, if you look at the cccDNA and RNA expression here, entecavir, which should not impact the size of the cccDNA pool, or the amount of viral RNA, but should prevent viral DNA from being secreted into the supernatant, actually does exactly that, which suggests that this platform can be used to actually look at the mechanisms of action of these antivirals as well. So the last thing that we wanted to look at here was just what exactly the spectrum of natural variation across different hepatocyte donors is in permissiveness to hepatitis B virus infection. And what we're able to see here by testing a number of donors, a subset of them is shown here, is that there are large differences in basal permissiveness to infection, but also in the response to JAK-STAT signaling inhibition. The other interesting thing is that this infection efficiency was actually not very well correlated to the overall hepatocyte health and function, which suggests that there are some potential genetic modifiers that we're able to capture a little bit with the system. The next thing that I won't have time to talk about today, but that we were kind of the first to show, is that as a complementary isogenic and renewable cell source system, we also developed an iPS-derived hepatocyte model of hepatitis B virus infection. Just as a review of what this first aim was, we were able to show that MPCCs do maintain liver-specific gene expression and properly localized hep B virus receptor over time, that we're able to use patient-derived HBV isolates to get robust infection and to show that there is actually an induction of ISG expression, and that inhibiting this induction actually leads to an increase in viral infection. So now that we had these two complementary systems, what we wanted to do was to actually use the primary hepatocyte systems to study the host response to viral infection and to use that to kind of perturb the viral life cycle. So to start out, it's kind of well known across a range of viruses, at least those that result in chronic infections, that they significantly remodel their hosts, partially as a way to persist and avoid immune clearance, partially as a way to remodel the host cell such that they become viral factories. And you can see across the range of viruses, there are a number of different pathways that get modified in this manner. For hepatitis B virus specifically, it's not a cytopathic virus, which means it can live for decades in humans and hepatocytes without killing the cells outright. And a lot of the host virus interactions that are induced by this are caused by the X protein, which, as I said, is a kind of promiscuous transactivator. This is just a subset of the different things that just the X protein has been implicated in affecting within host cells. There are things as broad as calcium release from the ER to a lot of transcription factors, including those that are liver enriched. Unfortunately, a lot of these experiments are done by just overexpressing one given viral protein, since the model systems are not exactly existent, and this may lead to some spurious results where you have non-physiological amounts of protein, issues like that. So attempts to more globally profile the host response to infection have led to data like this that kind of can swing from one extreme to the other. So people have looked at cell lines that either produce hep B or don't, and compare the gene expression profiles there. Unfortunately, these lines have diverged decades ago, and so are kind of difficult to really extrapolate from. And then work in chimpanzees has been subject to low N and also to variability that led to the inability to actually find differentially expressed genes and patterns of signaling that were induced by the virus. So what we know is that we need authentic host cells in order to really understand the gene expression profiles that are changing. And to just discuss briefly what the payoff is here, if we can really fully understand what the different pathways in the host hepatocyte are being modulated by hep B, we can maybe find points of intervention in order to actually clear the virus more effectively. So since we had primary hepatocyte model systems that can do this, we went ahead and performed transcriptomic analyses. So first we used single cell analyses of the transcriptomes of cells that were either infected with hep B or non-productively infected, and draw a diagram up to see how it feels to show one of these, but we're able to see by kind of an integrative analysis of all of the different signaling pathways that were modulated here that there are some interesting sets of biology questions that pop out. So one is that a core network of proviral factors that were annotated this way using pathway analysis, and you can see a lot of cells, a lot of the proteins that are circled in blue here are members of two heat shock protein families, which are kind of central to this regulation, which we, as we'll show later, actually intervened in. Another big thing that we found was that a number of proteins in the autophagy pathway were upregulated, signaling that perhaps the virus needs to actually remodel through the autophagic machinery as a way to get enough envelope to, say, bind the cell. Things like that. We also found, interestingly, the upregulation of some T-cell modulators, which our model systems can't really answer questions to, but are important, for example, in co-targeting for people like Anna, who were collaborating with an immune approach to clearance of the virus. Was to actually start perturbing these pathways. But when we went and wanted to use cell culture-derived virus as a more renewable source to try and perform these infection experiments, what we saw is that the infection efficiency was extremely low. So we saw about 2 orders of magnitude decrease compared to the initial inoculum, the amount of viral proteins that were produced. And so we went ahead and optimized the infection protocol by taking hints from a variety of other protocols in the literature, and we're able to see a 1 to 2 order of magnitude increase in both viral transcription and the production of which, when we looked for it, kind of luckily led to actually the understanding that we could see viral spread in vitro. And the first time that we were able to see, that anyone was able to see viral spread in vitro in HBV in these model systems. And we did this by reinfection after the inoculum was initially applied, inhibitor, and showing that the amount of protein and viral RNA kind of decreased. So now that we have this model system that mostly allow for cell culture drugged HBV replication, we went back and tried to ask a few other questions about interferon. And one that we were looking at, which is also kind of an unsolved question in the literature, is whether the virus blocks interferon signaling once those proteins are actually induced. And because we were looking at transcription, the way we looked at that here was to dose hepatocytes with interferon, which either were or were not infected. And so this was at a time where up to 2% of all the polyadenylated transcripts in these cells were viral transcripts. And even then, you see very little of a change in the response to interferon across all these different genes, across several orders of magnitude of induction. And us believe that at least transcriptionally and acutely, the virus does not actually block interferon-stimulated gene transcription, which means that the reason that interferon doesn't lead to viral clearance is probably not due to this. The other thing that I'm not going to be able to get into here, but what we found that is very important for the field going forward, is that model system differences are very important. So the results that we saw in culture-derived versus patient-derived virus actually diverged in some important pieces, and even the infection across hepatocyte donors was not extremely conserved, which shows kind of an underappreciated part of viral biology, which is that the host genotype really matters, and that taking these things into account with the model system that you can actually do that in is very important. But from there, we wanted to forge forward and actually look at whether we could target some of these pathways that had been implicated in the transcriptional data. And so what we did here was use an integrative approach to find pathways that were good targets of modulation based both on the centrality in those kind of networks that I showed you and based on the availability of small molecule inhibitors. And so when we did this, we were able to show with the example shown here are 2 classes of chaperone protein inhibitors and blocks autophagy, that we're actually able to get reductions in viral protein production and RNA production on mechanism that were even greater than, for example, interferon here. The fatty acid synthesis here, as I was told by a professor recently, actually just seems not to work because those small molecules don't work. Something. So overall, I think the main takeaways from this part of the thesis was that patient-derived HBV kind of remodels the hepatocyte in a way that affects liver-specific expression and to viral replication and to a viral infection. However, at least acutely inhibit the ability of hepatocytes to respond to interferon. But importantly, and this is something that can be extended even beyond hepatitis B, is these integrative analyses of the global transcriptomic set of cells and, under the conditions of infection can be used to nominate targets for intervention afterward. And that's something that is an interesting thing to take forward. So in the last part of the talk here, what I wanted to talk about was our approach to try and co-opt a novel genome editing system, CRISPR-Cas9, to directly attack cccDNA in the context of HBV infection as a novel way to try and treat the virus and actually develop a curative treatment. So a little more background on the current treatment of HBV here. So these nucleoside analogs and interferon were things that I'd shown before, are currently approved, but there are a lot of other types of treatments that are in clinical trials right now for Hep B infection. So there are entry inhibitors which try to block reinfection of new cells for the most part. There are other innate immune stimulatory ligands such as TLR7 ligands which aim to upstream kind of induce the interferon pathway and induce immune cells also. And then there's RNA interference which aims to degrade viral RNAs before they can produce viral proteins such that you can reduce the immunosuppression that's caused by some of these systemic viral proteins. Now, unfortunately, one thing that you'll see here is that none of these approaches actually directly target cccDNA. One thing that has been shown to target cccDNA is the innate immune response. So this is some data and schematics from a 2014 paper in Science showing that ligation of either the interferon receptor or lymphotoxin beta receptor can lead to the upregulation of these cytidine deaminases that convert Cs to Us in cccDNA, leading to degradation of this viral product. But unfortunately, as you can see, even with very high dose treatment for 2 weeks just in vitro, there's a trough of maybe 30 to 40% of cccDNA that can't really be cleared with this approach. So what we wanted to do was to look at having a more targeted approach. And just around this time, the CRISPR craze started. So I think this maybe won't need too much explanation to a lot of people here, but just as way of brief background, this is the genome editing system that was originally identified as a kind of adaptive immune system for bacteria as a way to prevent bacteriophages, which are viruses that infect them, from taking up residence in the host bacteria. And in the last 20 years, a lot of basic work has gone on these systems, but then in 2012 and 2013, the system was repurposed to actually work in human cells. It's an interesting note that it took until this was used in human cells for it to go viral. As you can see by the citation. But we were interested in it because this allows you to target the nuclease to DNA of interest, whether it's chromosomal or episomal, and it's kind of a robust, multi-flexible format that you can vector into either viral vectors or non-viral delivery platforms. So when we thought about CRISPR being used as an antiviral for HBV, we realized that it kind of satisfied a lot of the main conditions as a good target for this type of platform. So cccDNA as a target for Cas9, which is the nuclease, is episomal, meaning that when you're cutting it, you're not cutting into the host genome. It's a viral DNA, so it lacks a lot of homology to the host genome, which could lead to off-target effects. It's low copy number and in an accessible organ to novel therapeutic modalities such as RNA, because it's in the liver. The other important thing to note is that incomplete but high-level reduction can still spur immune clearance of the rest of the virus, and even reductions in intrahepatic viral load have been linked to a decrease in progression in some clinical trials that followed patients over several decades. So what we did to start this was first to select 24 guide RNAs, which are the RNAs that target to a specific DNA sequence across the HBV genome. Here, what we looked for were conservation across all the 5,000 full nucleotide sequences in GenBank, to tile across as much of the genome as possible and to minimize homology to the human genome as a way to avoid off-target effects. What we were then able to do is to test all of these guide RNAs just in transient transfection assays to look at the sequence context and look at which of these guides were most effective at actually reducing viral both protein and RNA products. So we settled on the 3 guide RNAs, 6, 17, and 21, since we didn't want to come up with better names for them, and then tested just in the early days to make sure that this would work in an in vivo context. This was using a hydrodynamic injection model of HBV infection that leads to viral protein and DNA production, but doesn't actually lead to cccDNA formation. So you'll see switch model systems later, but this was able to show that even in vivo, we were able to see pretty large reductions in surface antigen production and viral DNA secretion into the serum into the serum. So the next thing we wanted to do then is to actually look at a system that had true cccDNA formation that we could monitor over time and look at biologically what was happening to this viral DNA species as Cas9 was cutting. So here we use the HEP-G2215 system. Unfortunately, the names are basically things like that, where HBV is integrated into the host genome as a dimer and then leads to transcription and cccDNA formation from there. And then since we hypothesized that the overall potency of cleavage when you have these non-justifiably copy numbers in infected cell nuclei would be due to the total area under the curve Cas9 guide RNA complexes, we use a constitutive expression system by cloning a includes both the Cas9 and the guide RNA and stably transducing them into these cells and following them out over time. So what we see here is that we were able to get a pretty dramatic reduction in both total HBV DNA and in cccDNA. Reduction was progressive over time. So this is from 3 to 5 weeks out post-infection. You see going from about a 70 to 80% reduction all the way to 90 to 95% reduction. DNA formation, which you can see on the left here by qPCR, and on the right by Southern blot, you're able to see that these bands are almost completely gone. So that was pretty good data to us. Then what we wanted to look at was the actual mutational spectrum induced by these Cas9. And so we performed surveyor assays and next-gen sequencing, both on on-target and off-target locations. So what we saw here was that in the total DNA up here and in cccDNA down here, we're able, for the most part, to see high-level indel formation, which is indicative of mutations being induced by Cas9 followed by repair in these cells. One interesting thing to note here is that percentage indel formation in cccDNA was lower, and based on some other data that we've got also, we think that this is probably either due to this episomal template being cleaved and then just degraded because it's linearized now as a small fragment, or once it comes back together, not being able to really replicate. Those would be good outcomes as far as eventually getting this to be therapeutic. Then one important thing that we looked at here was whether we actually saw cleavage in any off-target sites. And we chose 8 computationally predicted sites and saw absolutely no evidence of cleavage. And this is not entirely surprising because our testing and trying to minimize homology to the human genome, for the most part, there were 3 and almost always at least 4 nucleotide mismatches in the 20 nucleotides of the guide RNA between any sequence in the human genome and our target. What we wanted to do was to actually test in a more true infection format. So here, now that those, at that time, the model systems had been developed to use the receptor knock-in in a hepatoblastoma cell line to get de novo infection. And so in models, both of cell culture-derived and patient-derived HBV, we were able to show similar efficacy, about a log or more reduction in cccDNA after transduction of this less strong vector. And also we were able to see cleavage of the cccDNA and indel formation in a system that wasn't tainted by this genomic DNA. So that was quite important to us. Which we learned as we were working through this, multiple other teams were doing the same things. They started to catch on to the fact that this would be an interesting tool to use against HBV. And so what I tried to do here was to synthesize all of the targeting to cccDNA cleavage in all these papers and compare that to nucleosome occupancy and histone context maps that were recently generated by scientists at Novartis across cccDNA genome HBV. And an interesting thing to note here is that although it does look like there's an enrichment in this area for guides that work, there are targets outside of that region actually be very dependent on nucleosome occupancy, which is just how many histones are there along with core protein, or on the histone methylation or acetylation state, which means that the best way to going forward actually find the absolute best sequences to use will just be to tile across the entire genome some regulatory sequences and overlapping read frames, which is something that's happening now. So overall, what we showed here was that Cas9, the CRISPR-Cas9 system, provides a platform that you can use to directly target HBV cccDNA for clearance, which is a novel mechanism of action compared to the other drugs that that people are thinking about using. That the informed selection of guide RNAs can lead to strong cccDNA cleavage and degradation without, importantly, genotoxicity due to off targets in the host genome. That I guess multiple companies now are taking forward because of the interesting data that a lot of groups have shown on efficacy here, and hopefully will be something that leads to part of the HBV armamentarium in a few years. So overall, I think to go back to just an overarching view of this thesis, the goal was to try and build tools and use the new technologies that are under development to get us closer to a comprehensive understanding of HBV biology that can be used to eventually develop curative treatments for the virus. So we showed the development of model systems using primary human hepatocytes for HBV infection, and trying to profile and perturb these responses in order to actually affect viral replication inside host cells and to affect the efficacy of this, and then to use the CRISPR-Cas9 system in order to directly attack cccDNA, induce clearance of this long-lived viral form. So that's basically the overview of the thesis. Now, of course, I get to the fun part. So if we had the room for longer, I could talk all day about how awesome it's been to work with all of the great collaborators and scientists in the lab as well. I think it'll suffice to say that I've had the pleasure of working with excellent scientists both inside and outside of the lab. Ram, thank you for being my committee chair and for maintaining a steadfast focus on really having impact with your science. I think that's something that's extremely important. Daryl, thanks for your insights, even though we didn't have too much time together on especially the immunology side of things. And both of you also thanked indirectly for a lot of stimulating scientific conversations I've had with people in your labs. And then, of course, thank you so much to Sangeeta for being someone who I can try to immerse with that picture. For being an excellent mentor, both in training my scientific eye to see what technologies and what science is the most important, for being so open with the lab and providing resources and enabling me to try to grow as a scientist personally, and then even for outside of lab things, being a mentor into the world of biotech, which has been exceptionally useful for me this entire time. So thanks a lot to all of you for that, and for allowing me to put some crazy pictures up. Thanks to the funding sources, and then to probably the most fun slide in everyone's defense. It's, I think, generally said that the middle years of grad school, for example, are kind of difficult before you can see the light at the end of the tunnel. Luckily, that hasn't been the case for me, mostly because of all of you guys who have been so awesome along the way. I definitely don't have time to thank everyone and tell you what you mean to me, but to family who have been awesome throughout this entire time. My cousin Sunil made it all the way here from to be here and provide these guys as support, I think. Thanks to everyone in LMRT over the years, even the people who aren't here anymore, to the boys, a lot of whom are in uniform, to the function crew, the Boston Hertz community / Gully, to Team Drones for Drugs, the IHST class, World Travel Buddies, and Clara, who even got me to buy new flip-flops. And a lot more people who I didn't mention here. But I can't wait to celebrate everyone else's successes and special moments as well. Now we can take some open questions, and I think after that you guys will get kicked out to go and do something that isn't being here.
Questions from the crowd?
I'll start off with the fun one. How did it feel to put up a hairball? You didn't say how it felt. So.
A little bit confusing. Tried to emphasize that the actual names weren't important and it was just the type of analysis that we did, which is.
You mentioned a few times how the genetics of the primary human hepatocytes would affect behavior in the system.
Mm-hmm.
What about the sort of environmental effects, like the BMI of the donor, or whether they're an alcoholic? What types of factors?
What types of things actually have been shown? So, interestingly, some of those environmental variables affect the expected kind of viral load, let's say in chronically infected patients, but as far as tying them to hard outcomes, such as the chance for progression upon, or for clearance or progression of functional cure upon therapy, there isn't actually extremely strong data there. There is some data, there are some SNPs through GWAS that have been implicated, although the odds ratio is all kind of below 2 in the chance of spontaneous clearance when you get infected. I think the reason for that is that it's kind of a binary issue whether you get clearance or not. And within that, there are modifiers that can affect the viral load, which is also what we saw in the in vitro systems, but nothing that will so dramatically change the overall.
So from like a public health perspective, is having this tool that attacks the CCC DNA directly sort of an important extra thing, or realistically from the perspective of cutting down on the number of infections that are currently out there, is it only like is it sufficient to have one or two of the therapies that you listed that are already in trials work out?
It's a question of what workout means, right? So in order to break the transmission link, you need to actually get clearance of cccDNA. So the chance of a baby who is born to an HBV-infected mother being infected, even if the baby is vaccinated immediately postnatally, is still close to double digits or low double digits, which means that you need to actually have a curative therapy for these patients in order to break the link to the next generation for transmission. Obviously, this is a novel therapeutic modality. Probably the fact that this is limited to the developing world is going to make paying for it, for example, difficult. But in Hep B specifically, one of the reasons that, for example, the RNAi players are so focused on this as a therapy, so Arbutus, Arrowhead, and Alnylam, is that it's such a large market that even if you were to price at a price that was affordable, say to China, you could still do very well. And so as far as adoption of the therapy, if it actually works out, I think that it's pretty positive. Yep.
So for cccDNA, did you try multiple guides together, or did you keep to one?
It's not because of off targets. It was more just for logistical reasons, and we wanted to see what the outcomes were from one. That is one thing I didn't show data. We have some data on multiplexing and how that actually definitely speeds up the reduction in viral, both protein and DNA, but potentially can lead to qualitatively different outcomes too, because you can kind of excise part of the cccDNA and definitely prevent it from rejoining.
Regarding your Cas9 experiments, did you observe sort of mutational escape of the virus from the cut sites?
Yeah, so we did not, partially because some of the regions that we were targeting were with overlapping open reading frames. So that's one thing. The virus is so small that when you have these overlapping open reading frames that are often not even in frame to each other, even mutations that may in one frame be synonymous, for example, due to indel formation or due to mutation at that site, won't lead to escape. And that is one thing we're going forward. One potential may be to look at actually RNA motifs that are important there, so like the signal sequence and things like that, where even synonymous mutations would be completely deleterious for the virus.
Questions? Going once? Okay, let's thank Vyas again.
So we'll ask the public to adjourn, and we'll meet in closed session.